name is Edna Casillas.
And today we're presenting to you some really awesome research on whole organ tissue engineering, decellularization and recellularization of extracellular matrix scaffolds. To begin with just some statistics, the American Heart Association reported in 2010, 1 in 9 death certificates mentioned heart failure. Of those, 20% of them found the heart failure as an underlying cause. Also, 425,000 annually people were diagnosed with coronary artery disease as well. Patients who die waiting for heart transplants is actually growing exponentially. Unfortunately, those who actually get to receive transplants have to deal with chronic rejection, immunosuppressant burdens, stuff like that. The demand for transplants, though, far outnumbers the number available.
Our major researcher for our presentation is Doris Taylor. She is the director in the tissue of engineering field and has a PhD in pharmacology at the UT Southwestern.
Not mechanical, but we're almost there.
Doris Taylor is building a heart from stem cells. Her process begins with a donated organ, which she then turns into a ghostly corpse.
What we're looking at here are rat hearts going through the decellularization process. And you can see here we have a heart that's still red and muscular. You can see one here that's partway through the process. And then here you can see a heart that's lost all of its muscle. If we sliced the heart in half, the valves would be there, the blood vessels would be there, all the rough inside lining of the heart would be there, but without cells.
Doris's goal is to transform heart transplants. She wants to seed a ghost heart from a donor with a recipient's stem cells and then restore it to life.
If we can use your stem cells to build you an organ, then you're not trading one disease for another like you do today. Today, you may get a heart, but you have to take anti-rejection drugs for the rest of your life. We'd love to be able to build an organ that matches you, is available for you, and that wasn't even fathomable 10 years ago, 15 years ago.
Okay, let's begin. The process of extracellular matrix scaffold. First, we will have to know what's this. The extracellular matrix scaffold is a substance produced by cells and secreted into an environment where the cells are embedded in a temporary platform or framework. Okay, the mechanisms by which natural scaffolding are promote for functional tissue reconstruction, but they're yet not fully understood. The ECM is definitively a differentiative pathway. It gives us a differentiative pathway in the understanding and the processes of human embryonic stem cells and some selected progenitor stem cells. The structure is highly complex, and therefore, when processing the biological extracellular matrix scaffold, they need to inhibit some bioactive peptides.
Next, we're going to talk about the actual decellularization process. Decellularization basically just means to remove all the cells of an organ while retaining the native composition and structure. This is done by exposing it to detergents, proteases, which are enzymes that break down proteins, and also by mechanical degradation, just agitating it. The ultimate goal in this is going to be to remove all the cell material without adversely affecting the composition or the biological activity and the mechanical integrity of the three-dimensional matrix. This first figure on top just kind of gives a separation of the different methods they would use to decellularize an organ. In our case, the heart, this bottom image you see here, this is just standard protocol. Because the heart has to be completely decellularized, they use a combination of processes. So you can see that there at the bottom. And then this is what it would look like during that process as well.
After the organs have been decellularized, scientists have seen the difference between male and female hearts.
One more quick video for you.
It's unmistakable.
If you can be a little wider and do the two shot of her holding.
The producer on this piece was Shari Finkelstein.
I bet most people, if you said one is male and one is female, which is which? They would say, oh, the soft, squishy, floppy one is the female one and the tough, whatever one is the male one. And yet, it's the exact opposite.
See, this one holds its shape so much better. That really did surprise me.
You would have guessed the opposite?
I would have thought they were more similar, I guess.
Her theory, although she doesn't know this to be true, but her theory is that the increase in how much blood the woman's heart has to pump during pregnancy would require a stronger structure.
Turns out it's true for every organ, kidney, liver, heart, the structure of our major organs.
The underlying
Are
framework is
seriously different. Now we're realizing that we are different down to our very cells. Our organs are different, our metabolism rate's different, our skin is different.
I have to say, I thought to myself, are those differences meaningful in a way that would affect how my mother or my daughter is treated when they go to the emergency room?
Well, it's a problem because while the researchers are discovering the differences, the medical establishment as a whole hasn't necessarily caught up.
Do you think that scientists, given what you now know, should consider sex differences in virtually every single biomedical experiment and clinical trial that there is?
Absolutely.
Every single thing.
Absolutely.
Therefore, scientists hope to use pig and human hearts in transplants because they have the same structure and size.
Okay, for this article and all these experiments, they needed some cell sources for the whole organ recellularization. We have three main cells. Both of them are allogenic and one is autologous. We have the embryonic stem cells and the fetal cells that are allogenic, and our adult-derived stem progenitor cells that are autologous. The embryonic stem cells are pluripotent and they are more programmable because they lack of epigenetic modification. This means that their genome is not yet modified, so they're mainly intact. The fetal cells are from the amniotic fluid and the umbilical blood cells, and they're more primitive and they allow more of a genetic mismatch. Both of these come from the umbilical cord blood cells, which means they're rather fetal than from the mother, than maternal. The autologous one, that is the ADSPC, the adult-derived stem progenitor cells, are from any tissue after birth, and there are known as the stomal cells. They are also, they cannot spontaneously give rise to any complex tissue in vitro, and then most none of these ones are the bone marrow cells that they are used in 600 clinic trials at the present. And
next, we're going to talk about how they actually recellularize these. They've got three methods of doing it. One is aortic infusion, retrograde aortic infusion, basically backwards infusion through the aorta. And the second one is brachiocephalic artery infusion. The third is a combination of inferior vena cava plus brachiocephalic artery infusion. And we have a model here just kind of demonstrating how they would do that. For our demonstration, this is actually the carotid artery. It would normally be in this one right here, which is the brachiocephalic artery. They found that to result in whole heart distribution of the rat aortic endothelial cells, that the brachiocephalic artery and inferior vena cava were the most proficient in that. The whole heart was proliferated, it retained its endothelial phenotype, and it expressed the nitric oxide and von Willebrand factor needed for clotting factors.
there are some cultural aspects for this. First of all, we have the reactors, which are the extracorporeal organ perfusion devices. We have three characteristics for this. One is that it has to have the normal body temperature. The second one is that it needs to be a continuous perfusion. It cannot be like, you cannot like stop and then start again. It has to be continuous. And the third one, it needs an oxygenator. This means that it has to have enough oxygen for it to work. And we also have the biophysical stimuli. The example for this one is the electrical impulses that it needs.
In summary, the most complex organs have been made from decellularized organ matrices and animal cells. If organs are to be truly capable of rescue or repair, they must function as a physiological enhancement or replacement of the damaged tissue.
The only downsides to this is that they've only successfully built these hearts to last a short term, just a few hours. So the next milestones would be to have them sustained for midterms, which is days to weeks, and then long terms, months to years, so they can actually use them for transplants. The problem is that vascularization is rare, so this limits the steps for tissue engineering.
Okay, organs such as heart, liver, lungs, and pancreas will have a failure if they present extensive fibrosis. Also, the adult human organs are often damaged, and every organ has a different tissue, so they are still making some experimentation to explore different targets for this. The whole organ tissue engineering is a potential breakthrough in medical science.
And these are all the sources that we use. We are happy to entertain any questions that you have.
I'm sorry, I was a little, can you just go over what your model was supposed to represent one more time?
Yes. This is obviously a human heart. This is how they would perfuse it. They would go in, this is actually the carotid artery. This is the artery they would go in, but for our demonstration to work, we had to do it through this middle one. This is the brachiocephalic artery right here, and then they would also infuse the inferior vena cava. We just didn't have enough clay to build that. But they would infuse it here, and it shows it washing over, and it's actually supposed to come out of both holes. But it would wash it over, and they would go through the, directly through the tissue. So this is basically our rat endothelial cells, and then we just perfuse them through the heart to show how it washes over and goes through.
Yeah, it has to go. It has to go through it and out of it.
So what's this going on? Is it chambers that they were
Yes.
saying?
Yes.
Yes. Okay.
And
Yes.
like she said, it has to have like a normal body environment, enough oxygen, normal body temperature. For it to actually function correctly, it has to mimic body function. So it has to have electrical impulses and stuff like that.
So those hearts in the chamber is representing a body, like a
Yes.
okay.
Yeah, it's a bioreactor, which she talked about earlier.
Wow. That is amazing. That's wow. Yay. Great subject.
More of the video, so I'm like, oh my god.
Yeah,
Yeah.
it's no. The second video is like 5 minutes long. So
Yeah,
I
it's
didn't have
awesome.
time to show, but it's awesome. It's really cool.
Now, okay, they were talking about different types of stem cells. There is something called induced pluripotent stem cell transduction, where they take the adult stem cells and then basically erase their memory of being an adult stem cell and convert it back to a stem cell.
Oh wow.
And what might be even better is to do the patient's own adult stem cells
Yeah.
and then erase the memory.
And that's what they're working towards. That's what they're working towards.
Yeah, but they having a lot of problems with the adult organs or
Yeah.
stuff because they're.
Because of the condition.
Yeah.
Mm-hmm. Yeah, that's the only downside.
And you would think they would just be like, oh, let's just use embryonic stem cells, fetal cells, but the problem is the whole
Ethical.
ethical
See,
part
that's
of
why
that.
induced pluripotent